hosting the RNAs collaborative seminar today from Würzburg. And without losing much time, I would like to start introducing our first speaker. We have two talks. The first speaker will be Redmond Smith. He's a professor and head of the Helmholtz Young Investigator Group in Würzburg, Helmholtz Institute for RNA-based Infection Research Institute in Würzburg, and he's a professor at the Medical Faculty of University of Würzburg as well. Redmond carried his undergraduate at the University of Cambridge in UK, specializing in virology and immunology. Afterwards, he moved to Melbourne at the Burnet Institute for his PhD, where he investigated mechanisms of HIV genetic diversity in natural host cells. Then for his, afterwards he moved to, for his postdoctoral research training, he moved to RNA research to Strasbourg. And in 2015, he was recruited by CNRS as a investigator, principal investigator. And in 2018, with Helmholtz Association, with the support of the Helmholtz Association, he moved to Würzburg and started his own research group at HIRI. So today's talk will be about probing structure and function of viral RNAs. And I would like to invite Redmond to share his screen now. Let me make a quick announcement for the questions. Please use the Q&A. And after the talk, in case we don't have enough time, I'm going to read them aloud and Redmond will answer this. Questions. In case we don't have enough time, we will move the rest of the questions to the after both talks, and we'll keep the Zoom open for that. So could you switch the window, Redmond? Can you move to a presenter view?
Can you not see the screen?
We need to switch the displays. We are
This
seeing
one?
the presenter view right now, and you need to move down down the screen.
What do you see?
There is I can still see the files or no.
Okay, yeah.
Right. Okay, would you try again?
I will try this.
Perfect. Yes, thank you.
Okay.
It's working and we can hear you. Thank you.
Okay, hello everyone. Neva, thank you for this really kind introduction, and to, of course, the organizers for the opportunity to speak today. So my name is Redmond, and it's a great pleasure to share some of the data that we've generated over this past few years. So as you might have guessed from the title of my talk, my group works on viruses, in particular RNA viruses, and we are trying to understand how these pathogens exploit RNA structure within their genomes as part of their life cycles. So today I'm going to give you a short introduction about my lab's interests, and then I will tell you about how we're probing the structures of long RNAs in order to understand their biology. So to do this, we actually had to develop an entirely new RNA structure probing technology that we're calling Nano DMS map, and this is what I'll present to you today. So within my lab, we have three RNA-centric research themes. So in the first case, we're trying to discover new regulatory motifs within viral genomes. So basically what we do is we determine their structure, we investigate their mechanisms of action as well. So in other words, we're trying to elucidate RNA structure function relationships within viral RNA with the ultimate aim of identifying conserved druggable targets. So in the second case, we're interested in RNA structural heterogeneity. And as you are all aware, many biologically important RNAs don't fold into single uniform structures, but exist as structural ensembles, with each structure having a distinct biological function. And we are devising ways to experimentally access this structural information, which is a very important yet still quite difficult problem in RNA biology. And finally, what we're trying to do is understand the role of genome architecture in virus evolution. So many RNA viruses use these fairly complicated mechanisms to assemble their genome into viral particles because it offers evolutionary advantages via genetic mixing. And we want to learn about the RNA dependence of these processes in order to be able to manipulate it for therapeutic purposes. So the story I want to share with you today relates to this idea that RNA sequences, single RNA sequences, can adopt different folds. So the classical example of this is ribose switches in bacteria. And these are RNA molecules that bind small ligands, resulting in structural changes that then go on to regulate gene expression. For example, by refolding a transcription terminator hairpin or changing the accessibility of a ribosome binding site. So a less prominent example of this phenomena, although probably more widespread, relates to alternative RNA structures found within different RNA isoforms. So these RNA isoforms can be generated through alternative transcription start sites or termination sites, as well as, of course, through alternative splicing. And by definition, these different isoforms will contain common regions of shared sequence identity. However, because RNA folding is contextual, that is, the folding of any particular RNA sequence is influenced by the upstream and downstream sequences, the RNA structures at these common regions are not necessarily identical. So we became interested in RNA structure function relationships in RNA isoforms because of our work on HIV-1. So many of you probably know this already, but HIV-1 is a retrovirus. This means it has an RNA genome, which is then reverse transcribed into double-stranded DNA that's then integrated into the host cell chromosome. And from this point on, HIV largely relies on the host cell machinery to transcribe and translate its genome. So the genome is actually transcribed into three major classes of RNA. So the unspliced, the partially spliced, and the fully spliced RNA. So these fully spliced RNAs behave just like cellular mRNAs and are exported via the classical pathway. And these single spliced or partially spliced and unspliced RNAs must be exported via a viral protein binding to an RNA structure within the genome, the so-called Rev-RRE interaction. So what's interesting about this unspliced RNA is that it has two fates within the genome. So first of all, it's packaged into virions as the genomic RNA. And secondly, it must be translated into the structural and enzymatic proteins of the RNA. On the other hand, these partially spliced and fully spliced RNAs are only translated and they're actually excluded from the packaging process. So this becomes important when we take a closer look at the genome organization of HIV-1, and we see that it's actually quite complex. So from the unspliced RNAs, we actually express two genes, GAG and GAG-POL. So with the GAG-POL protein being expressed thanks to a -1 programmed ribosomal frameshifting event, which is dependent on RNA structure here. And the remaining proteins are expressed from alternatively spliced transcripts. So we have one class which is partially spliced, transcripts of approximately 4 kb, and they express VIF, VPR, ENV, and VPU, whereas the fully spliced transcripts, which are approximately 1.8 kb, express TAT, REV, and NEF. So these three classifications are actually simplifications, because in reality, the virus makes use of a large variety of major and minor splice donor, as well as splice acceptor sites. And I've given you a few examples here, but in infected cells, over 50 different transcript isoforms have been detected. So as previously mentioned, all of these transcripts are efficiently translated in the cytoplasm into viral proteins, yet only this unspliced RNA makes it into virions. And this was my particular interest and puzzle for a long time, because in actual fact, all viral transcripts, including the spliced transcripts, share a portion of the 5' UTR, which includes the major packaging signal for HIV-1. So this is really the business end of the viral RNA. So this stretch of nucleotides is about 330 nucleotides long. It's highly conserved, highly structured, and these structures are really key for nearly every step in the HIV-1 life cycle. So if one is really interested in complex RNA function, it really doesn't get any better than this. And hopefully you can appreciate from this diagram the multiple kind of independent functions that are packed into this 5' UTR, sometimes on overlapping structures. For example, we have TAR here for transcription, poly-A for polyadenylation. This PBS binds a cellular tRNA used to prime reverse transcription. This stem loop 1 is very important because it contains a loop sequence that helps the genomes to dimerize, which is important for packaging. Important here also is the splice donor site within SL2, And then historically, SL3 was considered a major packaging signal. So the structure of the 5' UTR itself is subject to significant debate, although there is consensus that the 5' UTR acts as a ribose switch, where one fold remains monomeric and is preferentially translated, whereas the other undergoes dimerization and is packaged. And this dimerization, as I mentioned, occurs via SL1. So over the years, we've obtained evidence that the idea that SL3 is the major packaging motif has been displaced by this idea that SL1 is the major packaging motif. So whilst the mature NC portion of this protein binds with high affinity to SL3, it's actually the immature PR55 GAG protein, which is involved in the packaging process. And we've shown in a number of studies that this is recognizing the SL1 stem loop. So historically, we had this nice mechanism where SL3 was the packaging signal, and this was removed during the splicing process. However, if the major packaging signal is SL1, it's, as I've mentioned before, it's actually present in all spliced viral RNAs. So herein lies the question: how is HIV discriminating between spliced viral RNA and unspliced RNA? So we reasoned that a likely explanation for this selection against the spliced viral RNAs is if the structure of SL1 was somehow different in the spliced RNAs. So to investigate this, we turned to a technique which we use lots in the lab called chemical probing of RNA structure. And the principle behind this technique is very simple and indeed was highlighted in a previous presentation by Yue Wan's group in Singapore. So the idea is that we have some chemicals that react preferentially with single-stranded RNA rather than double-stranded RNA. These positions of modifications can then be read out in experiments. So most commonly, this is done by reverse transcribing your RNA and read out via next generation sequencing. So this is a very powerful approach, of course, for determining the structure of even quite long RNA molecules. But there is actually a major caveat to this process, and this is the fact that these short reads cannot be always unambiguously mapped to different transcript isoforms, such as those generated through alternative splicing. And as I've just told you, this is particularly problematic for HIV-1 because it has a complex splicing landscape. So if you were to map these data to a single transcript, what you would find is that you obtain a mix or an average of the different structures across the RNA. And of course, this makes it either difficult to interpret the data or impossible to interpret the data, depending on your question of interest. So the obvious solution to this problem is to use long read sequencing in order to map these reads unambiguously to each transcript isoform. And this is what we have done by developing nanopore dimethyl sulfate mutational profiling, which is a bit of a mouthful, so we call it DMS, Nano DMS map for short. So essentially, we're using the chemical probe DMS, which measures strandedness at A and C residues, and these positions of modifications are then read out as mutations using long read and nanopore sequencing. So although this may seem like a fairly obvious strategy, at the outset, we didn't really know whether this was going to work for two main reasons. So in the first case, we didn't know whether it was possible to obtain long cDNA reads containing mutations across the RNA. And this is because reverse transcriptases in general don't like reverse transcribing modified RNA, and they tend to fall off. And the second issue is that RNA structural profiling using mutational analysis depends on being able to see the signal from the mutation induced by the probe from the noise of the sequencing device itself. And as many of you are probably aware, nanopore sequencing does not have the same error profile as Illumina sequencing, so the error rate is usually much higher. So the first problem we solved using a highly processive reverse transcriptase called Marathon RT, developed by the Pile Lab. And she has shown that this enzyme is much more capable of performing error-prone bypass of modified nucleotides. So the second issue was initially much more problematic, as you can remark from this graphic here, that there was very poor signal to noise with increasing DMS concentrations if you consider the read straight up the sequencer. But what we were able to do was to examine the error profiles of the machine, of the data, and we were able to implement some reasonably simple filtering steps, which gives us really great signal to noise, even at low DMS concentrations. So what we're obtaining now is an apparent Phred score of Q30 or 99.9% accuracy, which is very close to what you obtain from Illumina sequencing data. And just to give you some insight on this filtering step, we noticed that the largest reduction in error is if you ignore indels. So this is obviously a very high error frequency in nanopore sequencing data. So what we did next is we performed a benchmarking study on a panel of highly structured and well-characterized RNA that we transcribed and then folded and tested in vitro. And what we do is we perform an analysis to test the match between the DMS reactivities obtained in the Nano DMS experiment with the strandedness of the RNA as determined by cryo-EM and crystallography. And what we see on average is we get a ROC AUC score of 0.8, indicating a very, very good match between the DMS signal and these known structures. And if just to give you a more visual example, this is the match between the DMS map signal and the RNA structure for the bacterial RNase P. So you can see the RNA secondary structure here. And on the right, you can see the Nano DMS map structure. So you can clearly see that nucleotides with high reactivities are classified as single-stranded, and nucleotides with low reactivity are double-stranded. So you can see here in red the loop regions, and in blue the single-stranded regions. So a more challenging benchmark we also performed, which is to determine the structure of the ribosomal RNA as probed in cells. So this is the 18S ribosomal RNA, which is 1.9 kb. We were able to sequence this in its entirety, and you can see very good correlation between the Nano DMS map signal and the regions of strandedness. And you can see here that we get very nice ROC AUC signals. So interestingly, the signal at the C residues is much higher than for A residues. So the C residues are often found in these double-stranded regions where there are many A residues in these loop regions. And many of these loop regions actually show protection against DMS reactivity. And we've seen this actually in other data sets online using chemical probes. And we think the explanation here is that there are RNA binding proteins at many of these sites that are blocking the DMS signal. And this is actually an advantage because we can perform experiments in order to identify protein binding sites on RNA, as you will see later. So turning back to our question at hand, we initially looked at the 5' UTR structure in the genomic RNA. We performed de novo prediction of the RNA structure based on our DMS reactivity, and we recover in this experiment the canonical or known structure of the HIV-1 genome as defined by Kevin Weeks's lab using shape chemistry. So as you can appreciate in this panel on the top right, we have very, very low error rates in our data when we don't add DMS, and we see strong increases in mutation rates at A and C residues as expected by DMS. And these reactivities correlate really well with strandedness on this structure. You might also remark on these slight increases in mutation rates at both G and U residues with increasing DMS concentrations. And there is an explanation for this. So classically, DMS reacts with this N1 position on A residues and N3 position on C residues at the Watson-Crick phase. It also reacts with the N7 position on G residues, although this does not give secondary structure information. Kevin Weeks's lab has recently reported that DMS can also, under certain conditions, react with the N3 position of the U residues, as well as the N1 and N3 positions of G residues. So we checked whether mutations at these residues were also able to correlate with RNA secondary structure. So whilst we saw no correlation with the G residues, indicating we're probably mainly seeing the mutational signal from this position, when we check for U residues, we actually get very high quality information despite the signal to noise not being great. And I think this really speaks to the power of Nano DMS and the filtering steps that we've implemented. So again, good separation between single and double-stranded RNA, and we get very good match between the signal and this RNA structure. So we have sensitivity, at least at high DMS concentrations and high coverages, to probe 3 nucleotides, which may facilitate RNA structure prediction. So we went on to explore this relationship in more detail, the relationship between DMS concentration and coverage. We did this by performing subsampling of our data sets. And what you can see here is that at high DMS concentrations and high coverages, the data quality is really exceptional. But we can also recover very good structural information at high DMS concentrations with only 1,000 reads. And when we have low DMS concentrations, very good quality data at 10,000 reads. So this is a quite modest read depths, and this is really quite achievable with today's nanopore sequencing. So as a final validation check, we wanted to see differences between the cellular RNA and the virion RNA. And here you can see reactivity information for the 5' UTR of the genomic RNA in cells at every concentration of DMS that we tested. And hopefully the first thing that you remark is the reproducibility of the signal. It's very good across different DMS concentrations. And when we perform a comparison between the signal found in cells and the signal found in virions, we see clear protections from DMS modification at 2 positions. So here you can see at the tRNA primer binding site and the loop of SL1. So in essence, what we're seeing here are RNA-RNA interactions important for viral replication. So in virions, the tRNA is loaded on the genome and the genome is dimeric. So this phenomenon of these is well known. However, we see a number of additional interactions that point to new biology. So for example, we see here in the poly A stem, we see protection from modification at this position, similarly here between U5 and PBS, this region downstream of PBS. We also see changes in reactivity in this tRNA-like element. So we can do an interesting experiment here where we deprotonate the RNA with proteinase K before doing the probing to remove the proteins. And what we see now is recovery of that DMS signal. So again, the reactivity reappears. And this is evidence that at these positions in the RNA, we have probably proteins binding that presumably have a function in viral replication. So now we're going to return quickly to the question at hand. What's the structure of the 5' UTR and spliced RNAs? So what we did here is we performed as a proof of principle. We devised a strategy where we could selectively amplify either the genomic or the partially or fully spliced RNAs. So we amplified them as complex mixtures, and these complex mixtures were then taken and sequenced on the nanopore device. And this virtual gel here shows that there's very good visual correlation between the lengths we see on the input in the gels and what we sequence. So most importantly, from these diverse and complex mixtures, we're actually able to unambiguously map the long-read nanopore data to a diverse selection of spliced viral RNA transcripts. And here we can show that we're actually detecting about 60 different isoforms in these infected cells. So when we compare the relative abundance of each transcript with increasing DMS concentrations, we see some modest biases against the long transcripts. These are the VPR transcripts shown in red, but at all DMS concentrations, we're able to recover sufficient read depths for structural analysis. Another important thing to note is that between replicates, we actually get a very high correlation between the relative abundances. So the reproducibility of this data is really great. So now we have structural information in the form of DMS reactivities for different spliced RNAs. So the first thing we wanted to do was just simply look at the correlation between reactivities in the unspliced transcripts versus the spliced transcripts. And hopefully you can remark here that all of the spliced transcripts are very similar to each other and different to the unspliced RNA, whether it's in cells or in virions. We are also able to perform a clustering analysis, and we do see some subclustering according to first splice acceptor usage, in particular the A1 and the A2 splice site, and this is somewhat expected. So what I'm showing you now is the DMS reactivities for the unspliced RNA here versus a selection of different spliced RNAs, and the difference between the two is shown as a graph on top. So we see some expected changes due to the loss of downstream sequences that are only present in the unspliced RNA. For example, here we see a dramatic increase in reactivities in the spliced RNAs because we no longer have this downstream sequence which forms a pseudoknot. We also see the loss of the U5-AUG interaction due to the fact that this sequence is spliced out. But we also see two changes that were less expected. Here we see evidence for remodeling of the primer activation sequence in a consistent way in all spliced RNAs. And most importantly, we have very strong evidence that SL1, the stem of SL1, is not formed in any of the spliced RNA, and this can be observed as strong increases in reactivity to the DMS in this region. So what we think is going on is that splicing is removing this 3' portion of the stem, and the 5' portion kind of then snaps back onto SL1, destructuring SL1. And we also think that this might be helped by this region in PBS, and this is an interaction that we identified and published this year. So this is presumably a nice mechanism for how HIV ensures its spliced RNA is not packaged in a way that's kind of independent of the splice acceptor usage. So this is the model that we're working towards. However, we do need to be cautious because so far the DMS supported modeling doesn't really give us satisfactory results. And I can give you an example of this. So here's a predicted folding of a D1A1 spliced RNA. So we see some interactions between this SL2 region and the downstream sequences. And this folding is kind of consistent with the DMS reactivities. However, in this region where SL1 is present, we see base pairing predicted that's actually not supported by these very high reactivities in the SL1 stem. And this really reflects a limitation of the RNA folding algorithms that we have available to us and not the quality of the data. And moving forward, we'll need to perform and to use more advanced RNA structural probing methodologies, such as two-dimensional probing. So just before finishing, I want to highlight that it's possible to analyze very long RNAs with Nano DMS map. And we've actually probed the entire HIV-1 genome in 2 different amplicons. So here we have very nice correlation between the reactivities obtained in our data versus shape probing data sets. And in this bird's eye view, you can identify already very highly structured regions, such as the 5' UTR, the frameshift element, the rev response element. And it's also fun to see how RNA structural probing has advanced over the years. So in this landmark paper, the first paper from the Weeks lab, they determined the structure of the 5' UTR using 19 liters of supernatant and 31 primer reactions. And here we do it from 2 ml of supernatant and 2 PCRs. So we get, and these are just examples to show you that we get very good correlation with known structures from this long-read sequencing data. And finally, because these experiments are so simple to set up, we can rapidly perform, for example, footprinting experiments across the entire HIV-1 genome. And here you see some preliminary data where we deprotonated the viral RNA before probing. And interestingly, you see decreases in reactivity upon deprotonation, which may indicate that, for example, the viral MP protein that coats the viral RNA is holding open RNA structure, which may be important for later steps of the viral life cycle. This, of course, needs to be followed up, but it's just to give you an idea about some of the experiments that are possible with Nano DMS map. So in summary, I've shown you that this is an easy and robust method to probe the structure of long RNA molecules. And we also use this to show that the structure of the 5' UTR and the spliced viral isoforms is structurally distinct from the genomic RNA. And we think this explains why these RNAs are excluded from the packaging process. And with that, because I'm out of time, I'd like to thank the 2 people who performed the vast majority of this work, Patrick Bond, who's a PhD student, and Anne-Sophie Grebling, who is a postdoc, and to all of the funding agencies, and to you for your attention, and hopefully there's time for questions. Thank you.
Thank you very much, Redmond. So I'll try opening my video as well. Doesn't work. Okay, we'll start with the Q&A. So we have already some questions popping up in the question and answer window. So first one is by Shashi Singh. How are RNA maintaining the two structure in same environmental condition, even though they have same sequence as you have shown in spliced and unspliced RNA?
So we think that, yeah, if I understand the question correctly, that there's an intrinsic ability for this sequence to fold into alternative structures, and there's an equilibrium between the two. Of course, that equilibrium can be pushed in one way or another through, say, the binding of host or viral factors, and we showed this in the NSMB paper, that we think that the binding of the tRNA can actually help the RNA become dimeric. So this is the 5' UTR in the genomic RNA, but of course the same principle applies in the spliced RNAs.
Another question by Vanessa Mondol. Does this require custom modifications, oligos to the nanopore sequencing kits?
No, so we're using the nanopore kits that are available. So we used for this study the updated flow cell chemistry, so the Q20 chemistry, and this makes a huge difference. So you really need to do this to get the best quality data. As for the oligos, you know, there's a variety of different methods to prep the RNA or the DNA for sequencing, and these are all available from ONT. Mm-hmm.
And I have a quick question, Redmond. In your benchmarking, you have mentioned you checked the structure from cryo-EM and the DMS, previous DMS experiments. So do you see your data correlating well with them, or does the both data, cryo-EM and DMS from previous studies, also correlate well with?
Yeah, so the panel of RNAs we picked from a Riju Das's paper that determined all of these structures by cryo-EM, but they were already determined, a lot of them by RNA crystallography, for example. And yeah, we get very good matches between the DMS data and these structures. So these structures are very hard to fold to de novo. So so it's nice to do this via benchmarking, where we know which regions are single-stranded and which are double-stranded, and that allows us to do these ROCA-UC calculations.
Excellent. Another question by Rafael Hasleka. Could you also detect or differentiate between DMS modified M5C or M6A on the RNA?
Okay, yeah, that's a really interesting and great question. I think in principle, this is possible. So the data that I already showed you in the presentation, one of the modifications that's induced by DMS is this N7G modification. And we see in the data that this appears to have a very unique mutational signature. So I think we only see G to A mutations. And this type of approach can apply to other modified bases. So Anna Pyle's group has done some work on this. She calls it ModSec. I don't know whether the paper is online yet, but you can send her an email and ask her about it. Of course, the other way of looking at this is to do the direct RNA sequencing.
Mm-hmm. And last question from Higor Pereira, and the other one I think you could answer just in the chat. Thank you for your talk. Just curious, were you able to identify long RNA-RNA interaction in the full genome of HIV, which might indicate a circular form?
Yeah, not directly, but that's something that I think might be quite likely. So we did some folding using the DMS data across the entire genome, which is something that you should never really do. I mean, there are possibilities for long-range interactions. It's not really the best approach to do this. There are other more adapted methods to detect these long-range RNA interactions. But in principle, you can do it.
Mm-hmm. Okay, thank you. Katrin Relly's question we will leave for after the session, or you could just answer it through the chat. So with that, we thank our speaker again, and we'll move with our second speaker, Bhupesh Prusty. So Bhupesh is a principal investigator at the Institute of Virology and Immunology at the Maximilian University of Würzburg. So Bhupesh started his PhD in University of Delhi and then moved to Germany for his postdoctoral training at the German Cancer Research Center. And for some time, he was also a postdoctoral researcher in Würzburg, University of Würzburg at the microbiology department. Then he, in 2018, started his own group under the department of Lars Doken at the Department of Virology. Bhupesh's research is focused on herpes virus latency and reactivation and roles of microRNAs in pathogenesis and much recently viral transcriptomics at the single cell level. So we look forward to your talk, Bhupesh. Could you please start sharing your screen? Great, we can see it in full screen. Thank you.
Perfect. Just a minute. I just have to move. Okay. Yeah. So thanks, Neva, for the kind introduction, and I'm glad to be here today. Hi, everyone. Today I'm going to share some of our recent work with you, which will mainly be focused on regulation of microRNA biogenesis by other microRNAs. So RNA is a very unique molecule which is often used as a tool by pathogens and hosts for their defense. Interestingly, RNA cross-kingdom transfer is not very uncommon. For example, fungi in particularly secrete small RNAs, including microRNAs, into their plant host and modulate plant cell biology. Parasitic plants like Cuscuta that grows on another host plant also secrete microRNAs into their host. Arabidopsis, for example, can exchange microRNAs among themselves by secreting them into the outside medium. It's not always self-defense. In a recently published PNAS paper, a nice example of symbiosis is also shown, where a fungi living around the roots of eucalyptus tree secrete a microRNA into the host plant root that allows establishment of beneficial relationship for both the host, that is the plant, and the fungi. Now, all these examples show how powerful can microRNAs and other small RNAs be that very often by itself is sufficient to manipulate the target cell. In these examples, pathogens were outside the host cell. The first line of host defense was the cytoplasm. Pathogens were clever to secrete the mature microRNA that would not be detected by the innate immune proteins within the cytoplasm. Now, what if the pathogen is already within the cell? And in this case, the pathogen is very often inside the nucleus, which is the other side of the cytoplasmic wall of defense. So today I'm going to talk about one such common viral pathogen which use microRNAs to manipulate its human host from within the cell. As you may know, our cells are home to numerous latent viruses. There exists a tight control over the function of these viruses within our cell. These viruses are kept dormant or inactive by various innate immune mechanisms. Some of these viruses even coexist with us for our entire life without causing any harm. One such virus is human herpes virus type 6. We all get infected with this virus at some point of our life during the early childhood. Even up to 1% of the human population inherit these viruses from their parents, thereby carrying 1 to 2 copies of the fully functional viral genome in every cell. Now, HHV6 is a unique pathogen as it has a telomeric repeat, TAACCC sequence at both ends of the viral genome, which allows it to integrate into the subtelomeric region of our chromosome. This specific location of the virus within our chromosome allows its frequent reactivation also. So how does this virus manipulate the host defense by staying within the cell nucleus? My lab specializes on HHV6. Now, when we created first ever reporter HHV6 cells carrying latent integrated HHV6, we wanted to characterize the very early stages of virus reactivation. In these cells, the reporter fluorescent cassette is within the viral genome, and it is silent when the virus is latent. But upon reactivation, the fluorescent reporter is expressed, making it easy to identify the cells with reactivating virus. Now, when we looked into the viral mRNA within the first 24 hours of reactivation, we detected a few viral transcripts. There were also not many changes in the host transcription profile, as you can see from these volcano plot. However, as early as 16 hours of applying the reactivation stimuli, we started to see several viral microRNAs in amounts easily detectable by northern blot. When we looked into the Argonaut-bound microRNAs, we also saw many of the viral microRNAs already within the Argo complex within the first 24 hours of reactivation. This was a bit surprising, as the viral DNA yet has not started replicating by this 24-hour time points. So what we did is we took U2S cells with latent HHV6A, because these cells use alternate lengthening of telomere. This is a mechanism for telomere maintenance, making it easier for the virus to reactivate from the telomere. We reactivated the virus using trichostatin A, which is a stack inhibitor. The viral reactivation is not productive here, as HHV6 only undergo productive reactivation in CD4 positive T cells. Now, upon virus reactivation in these cells, we saw a significant change in mitochondrial architecture. So you can compare the left panel with the right panel. The mitochondria starts fragmenting upon virus reactivation. The mitochondrial fragmentation was quantified. That showed increased mitochondrial numbers. As the mitochondria fragments, their number increases. And at the same time, there is a decrease in mitochondrial average surface area, because the mitochondria starts to become smaller. Now, the same phenotype could also be seen during the infection of primary human umbilical vein endothelial cells, or the HUVEC cells, with cell-free viruses. HUVEC cells allow the virus to grow and form resolvers within the cell, but they do not release the infectious viral particles. So here we see even stronger mitochondrial fragmentation phenotype. Now, one of the most common mechanisms by which mitochondria fragment is via the GTPase protein DRP1. DRP1, when moves from the cytoplasm to the outer mitochondrial membrane, it forms a ring around the mitochondria and fragments it. DRP1 is transcriptionally regulated by P53, which in turn is negatively regulated by miR30 family members. miR30 has 5 members, miR30A, B, C, D, and E. We observed the same upregulated P53 DRP1 excess during both lytic and latent virus reactivation. So you can see over here. In fact, we could also quantify the upregulated DRP1 rings around the mitochondria in the virus reactivating cells. Now, miR30 family members are the most upstream members in the P53 DRP1 axis. When we checked the transcription of mature miR30 family members, we saw downregulation of most of the miR30 family members of an HHV6 infection. But it became interesting to see that mature miR30 downregulation is not a transcriptional suppression of microRNA expression. Rather, it is a defect in the miR30 biogenesis, as primary miR30C starts accumulating within the cell, while mature miR30 starts disappearing here. Only miR30C is shown in this picture, as it is one of the major miR30 family members in CD4 positive T cells. Similar results were also observed from HHV6 reactivation studies in U2S cells. Now, microRNAs are first transcribed within the nucleus as a primary transcript, and then are processed in several steps using nucleases like DROSA within the nucleus, dicer within the cytoplasm, to finally produce a microRNA duplex, and then this is processed to form the mature microRNA. Several proteins are known to interfere in this process. One of the well-characterized protein is Lin28, which can interfere with the microRNA processing by binding to the hairpin loop region. Prior to our work, at least 3 other labs from around the world showed that human or mouse microRNA can actually bind to the same or different pre or prime microRNA and interfere with their biogenesis process. As I mentioned earlier, during the very early stages of HHV6 reactivation, even the viral DNA do not replicate, but produce any major viral proteins. So what might be responsible for this strong mitochondrial phenotype? We found that one of the viral microRNA called miR-A-U14 has the potential to bind to the hairpin loop of miR30 family members in a very sequence-specific manner. Viral microRNA is shown in blue in this upper figure. You can see here within the gray box that miR-A-U14 has a perfect sequence complementarity to the hairpin loop of miR30C. I showed you before that many viral microRNAs actually transcribed within the first 24 hours of virus reactivation. miR-A-U14 was one of them, and it was also found to be ago-bound. We used in vitro microRNA processing assays where a radio-labeled pre-miR30C was allowed to undergo in vitro processing using immunoprecipitated human ago2 protein. And we saw that a wild-type miR-A-U14 mimic can actually interfere with the processing of pre-miR30C and inhibit the formation of mature miR30C. Similarly, miR-A-U14 also has a perfect sequence complementary to the hairpin loop of miR30A, which is another member of the 30 family. And a miR-A-U14 mimic could interfere with the processing of pre-miR30A. Now, it is important to know that 30A and 30C constitute the major bulk of microRNAs from the miR30 family. As a proof of concept, we tested synthetic microRNA mimics. We compared the effects of a wild-type miR-A-U14 mimic, shown at the top, with a mutant counterpart having 3 nucleotide substitutions within the potential binding region. Indeed, transfecting cells with a wild-type miR-A-U14 mimic induced miR30C processing defect, as you can see here, as well as alterations in p53 and DRP1 axis, whereas a mutant mimic did not have the same effect. Quantification of p53 and DRP proteins from independent experiments are shown in these graphs. We could also show that wild-type miR-A-U14 mimic caused mitochondrial fragmentation, whereas the mutant mimic could not do this mitochondrial fragmentation. Now, the next step, we created a mutant HHV6A virus, which lacked the expression of miR-A-U14 using reverse genetics. From previous publications from Mori lab in Japan, we knew that U14ORF from the virus is very essential, and its mutant have severe growth defects. Hence, we did not alter the amino acid sequence of the U14ORF. This is just to mention that the microRNA AU14 is in the reverse strand of the UORF. But even without altering the amino acid sequence, our microRNA AU14 mutant virus growth was severely affected, suggesting a functional necessity of this microRNA in the virus life cycle. Hence, we generated U2S cells with integrated latent form of the virus that could be reactivated using TSA. Now, we looked into mitochondrial architecture upon virus reactivation in both wild-type and the mutant virus carrying cells. On the left panel, you can see wild-type virus reactivation. One can see mitochondrial fragmentation within reactivating cells carrying wild-type virus. The cells are green because the GFP cassette is within the viral genome and it is expressed after the virus reactivation. On the right panel, you can see that no major alterations in mitochondrial morphology upon reactivation of the mutant virus. Reactivation of this mutant virus did not show miR30 processing defect phenotype, which one can see in wild-type virus marked here within the red box. The characteristic phenotype of accumulation of primary miR30C is lost during the reactivation of the mutant HHV6. Now, we wanted to show that this interaction between HHV6 encoded miR-A-U14 and the miR30 is mediated by the hairpin loop. So what we did is we created 2 different artificial microRNA hairpin structures, miR-A and the miR-B, using lentiviral vectors. Each one carried a different sequence combination, but with the same exact hairpin loop that we find in miR30C. Additionally, we expressed either wild-type miR-A-U14 or its mutant counterpart in these cells. Under both conditions, we could show that wild-type miR-A-U14 was capable of inhibiting the maturation of the artificial hairpin loop, whereas the mutant miR-A-U14 did not have any effect. This confirmed the hypothesis that the potential interaction site of miR-A-U14 is within the hairpin loop of miR30C. Now, we then asked if this microRNA-mediated regulation of microRNA biogenesis is specific to only miR30 family, or we can create similar scenario for other human microRNAs. Interestingly, we observed that some of the human microRNAs, including LED7 family members, have an extended hairpin loop of up to 30 nucleotides. We chose one of them, LED7D, for example, here. We designed 2 artificial antisense RNA sequences that can bind within this loop of this LED7D, shown here within the gray boxes. When we transfected cells with these antisense RNAs, we could show that they interfered with LED7D processing, causing accumulation of the pre-miR LED7D. Similar experiments with another LED7 family members, LED7F, also showed identical results. Now, we asked if a pathogen-expressed microRNA can regulate a human microRNA. Why can't one human microRNA regulate another? I told you before that there is already literature available for such an example. We did some in silico analysis and found that actually a lot of non-human microRNA can bind to other pre and prime microRNA and can also self-regulate own biogenesis. But it was difficult to validate them experimentally, as majority of them are not expressed in a quantity that can be detected by northern blot. During this in silico analysis, we realized that microRNA can also bind to the other end of the stem loop of the pre-microRNA and can also potentially interfere with DROSA binding. One very clear example was binding of miR155 to the 5' end of miR148B. Both these microRNA are key players in immune response. Therefore, we transfected cells with miR155 mimic and observed the accumulation of primary microRNA 148B when mature 148B amount decreased, suggesting an interference in the maturation process. When we used radiolabeled primary microRNA 148B and immunoprecipitated human DGCR8 complex of proteins in vitro microRNA processing assay, we saw that efficient processing of primary microRNA 148B to pre-miR 148B happens. However, a wild-type microRNA 155 mimic severely interfered with this process, as intermediate accumulation of intermediate-sized products in the assay can be visible in these plots. These results point towards a more generalized mechanism of regulation of microRNA biogenesis through other small RNAs. We then asked if mitochondrial fragmentation can somehow suppress the innate immune response, which in turn can allow better viral reactivation. For this, we transfected 293 T cells with either wild type or 2 different mutant forms of the miR-U14 mimic, and 48 hours later exposed the cells to triphosphate hairpin RNA, which is basically a pre-Guy MAPS pathway antagonist that stimulates the interferon signaling pathways. Both interferon beta as well as interferon stimulating gene IFIT1 mRNA levels were comparatively decreased in 293 T cells, which were transfected with wild-type miR-U14 in comparison to the both mutant counterpart. This confirmed our hypothesis that the mitochondrial fragmentation through the miR-U14 is capable of decreasing the innate immune response. Most importantly, transfection of wild-type miR-U14 mimic into the U2 cells carrying a mutant HHV6 allowed latent HHV6 reactivation, even in the absence of a strong reactivation stimuli like TSA, as you can see over here. Interestingly, miR-U14 mimic was enough to cause miR30C downregulation, as you can see here, and virus reactivation, which is measured in the form of northern blot figures in the form of transcription of several viral microRNAs like miR-U2 and miR-U86. On the right panel, expression of viral miR-U86 is quantified from these northern blots. Now, in summary, hopefully, I managed to convince you how a pathogenic microRNA encoded by a common herpes virus regulates the biogenesis of host microRNA, leading to regulation of the p53-DRP1 axis, subsequently causing mitochondrial fragmentation that result in suppressed innate immune response, decreased interferon production, which we predict to be useful for the initial stages of the virus life cycle. With this, I would like to acknowledge all my lab members, entire Dolken and Erhard group, other collaborators and funding sources, and also to you for your patience. And now I would be happy to take questions. Thank you.
Great talk, Bhupesh. Thank you very much. All right, there are questions already in the Q&A window, so I'll start reading them aloud. Bastian From asked 2 questions. The first one, where in the cell, nucleus or the cytosol, is the proposed targeting of the human microRNAs by viral microRNAs are occurring?
I mean, the experimental data that we have done so far indicates that it is both nucleus as well as in the cytoplasm. There is no specific location for sure.
Mm-hmm. And another question from Bastian again. You propose that miR-AU14 targets the loop through a sequence complementarity in post 4/9 of the viral mature microRNA. Is this mediated by the AGO?
We believe that to some extent it is mediated by AGO, as we can show it by the in vitro cleavage assays. But also we believe that there is more to it. Probably other proteins are involved in the process, as we don't see. For example, when we did with the miR-155 cleavage assay, we saw very nice inhibition, but when we did with the miR-30, we did not see complete inhibition. So maybe there are other proteins involved.
Mm-hmm. I have a question, how I can second on that. Do you see that LED protein, which you said that binds to the loop region of some microRNAs, do you see that protein actually present in your microRNA together with your microRNA?
Which one? Sorry,
there was a protein called LED. You have
it's the microRNA LED7 family.
Okay, I thought you mentioned a specific protein binding to the loop.
Lin28. Lin28.
Yeah.
Yeah.
Do you see that on your microRNA?
No, we did not check it specifically.
Mm-hmm. Would be interesting.
Yeah.
Another question by Sophia Ademia. Does reactivation of viral microRNA simultaneously lead to reactivating human microRNA, or is it a subsequent process?
I mean, we saw the effect transiently within the first 24 hours. So we predict that as soon as the viral microRNA is produced, matured, the primary job of it to downregulate the miR-30. So it's going on at the same time when the viral microRNAs are appearing in the cytoplasm or moving to the nucleus. Yeah.
Mm-hmm. Wang Tang asks, says interesting finding. Can I assume that many mRNAs interact with pre-microRNAs? Do they affect pre-microRNA processing?
I do hypothesize the same. I personally believe that most of the infectious pathogens, particularly the DNA viruses and RNA viruses, they have probably evolved a way to manipulate the cell by producing the small RNAs, not necessarily the microRNAs. It can be non-coding RNA, small non-coding RNA, or large non-coding RNAs, and they all probably do the same thing to prepare the cells for the virus to survive and grow, so they can manipulate the things. So it is a ongoing research topic with us.
Mm-hmm. Vanessa Mondol asks, do you see primary microRNA miR-30 and AGO pull down after viral induction?
Yeah, we do see that, yeah.
Mm-hmm. Then a comment and a question, great talk, Sophia Ademia says, and do you consider those viral microRNAs as a master regulators of human microRNAs? Very interesting concept.
Yeah, so this is not one of the example that we have. We have another example from the same virus which regulates another family of microRNAs, human microRNAs. So we believe that the concept is not very one of a kind of example. It exists in a much more larger and broad form. We just have to find out more examples from it. As I told you, we have 2. Maybe there are more.
Mm-hmm, mm-hmm. Thank you. Shipra Malik is asking a question, can viral microRNA directly alter the expression of human mRNAs?
Yes, there are examples, this happening from previous literatures, yes.
Mm-hmm, mm-hmm. And I have a last question. So about the latency and the viral reactivation, you showed that viral reactivation leads to this microRNA mitochondrial fragmentation. Is that something commonly observed by other viruses? And you related directly to microRNA, but what if that means no human microRNA does such a thing? And do you actually see if from the latency in the first active phase, this must also happen, right? Before a virus goes into latency.
Yes.
And that should also lead to some fragmentation of mitochondria. So how do these cells would be basically lysed and dead. So in some cells, this means there must be latent virus without the, so some of the mitochondria should survive this, right? That's the question.
So the mitochondrial fragmentation is not the dead end to the cells. Mitochondrial fragmentation is a transient phase where the cell can persist for many, many days. If you keep the mitochondria fragmented in a cell culture dish for more than 5-6 days, then the cell starts to undergo mitophagy and they die. And mitochondrial fragmentation is also an essential part of generation of the mitochondria. The old mitochondria is generated by mitophagy through this process. So this is not completely bad for the cells, but the mitochondrial fragmentation for a prolonged time induce hypometabolic situations where ATP levels and the metabolic profile of the cell goes down. So it's some sort of like a winter sleep for the cells. Now, many herpes viruses are known to do the same thing, mitochondrial fragmentation, and the already known mechanism is through proteins, because they have evolved. So different viruses have evolved different ways to do it. And we believe that this is not one of a kind example. It is quite possible that we can see similar things because we see similar conservation of the same sequences within other herpes viruses also. So yeah, to answer your questions, it is quite possible that we can see mitochondrial fragmentation with many other viruses, and probably similar mechanisms are involved. Yeah. In
human microRNAs as well.
Yeah. This is something probably we can use Redmond's techniques in the future to see if RNA-RNA interactions between the pre and primary microRNAs and other microRNAs exist in larger scale, and which can tell us more information on the open regions of the pre and primary microRNAs, and that would be fascinating. Yeah.
Yeah, this is fantastic. Another question by Catherine Rellis, does viral reactivation induce immune response effects within the cells?
Sorry,
Does viral reactivation induce immune response effects within the cells?
No, the viral reactivation actually suppresses the immune response, so it doesn't induce immune response, rather suppresses it.
All right, thanks again, Bhupesh. We've been through all the questions. As I mentioned in the beginning of the session, if you have further questions, if you would like to ask directly more questions to both our speakers, please stay on Zoom. We'll keep it online for another 10 minutes, I guess. And with that, I would like to conclude the session. Thank both of our speakers, Redmond and Bhupesh, for these fantastic talks. And thank our participants for their interest. Have a great day.